before I do that, I wanted to make a comment about the exercise. So in the numerical exercise, you're asked to calculate the Lyapunov exponent, basically, roughly speaking, in two ways. One, actually numerically simulate two different trajectories that are nearby and calculate how far apart they are, the norm of their difference as a function of time. And the other is the way that we discussed in class, which was by basically calculating the Jacobian at a given point in space and calculating the maximal eigenvalue of the Jacobian. And what you may have already seen and what you will see in the exercise is that you don't get the two don't match. And so I wanted to just make a comment about that, clarify a little bit about that. One of the things also in the exercise you were asked to do is to plot the overlap between the eigenvectors of the Jacobian matrix as time evolves. And somebody just asked me if I really meant time steps of 1 over 10 milliseconds. And I really did. And you'll see that overlap decays within that time scale, within a single millisecond. The overlap between the eigenvectors of the Jacobian, the eigenvector associated with the maximal eigenvalue of the Jacobian, the overlap decays to 0 within that first millisecond. And so that's a kind of hint towards why it turns out that our calculation, meaning our calculation of the maximal eigenvalue of the Jacobian matrix, doesn't give us the real Lyapunov exponent in terms of the deviate divergence of the trajectories. And that's really because immediately as the trajectory proceeds, the direction of divergence is changing all the time. So there's not a coherent divergence along the path in which if there was a coherent divergence and the directions were stable, then those two eigenvalues would match. But at every single point, those directions are changing so fast that really the real numerical Lyapunov exponent requires averaging over space, which we ignored in our calculation. So that's that. Okay.
I don't know if it's related, but what I saw at the beginning, I simulated for longer time than you asked, and when I plotted the delta, delta x, it looked semi-linear, and then it became completely straight. And is it somehow the edit was?
Yeah. So also, if you look at the deviance, the norm of the difference between the trajectories over different time scales, also you'll see it's actually very hard. I had to kind of fine-tune the time scale which I asked you to simulate to find the time scale at which it actually looked like an exponential divergence. If you look at smaller time scales, it originally might decay first, and if you look at longer time scales, anything could happen. We know at the end of the day the divergence between trajectories is bounded. It can't be exponential in all time because the state space itself is bounded. We're talking about it bounds at plus or minus 1, so the maximal divergence of trajectories is inherently bounded. But yeah, it's really only kind of for a particular time scale that you can find the exponential-like behavior. Okay. So I wanted to, Jonathan asked me to talk about my research and the work that came out last year on the impact of structural heterogeneity on excitation-inhibition balance in cortical networks. And yeah, it's fun that it's relevant to the class. So it's a nice opportunity. And it's only one example of many ongoing works and studies of excitation-inhibition balance. So this is the outline of the talk. The introduction to balanced state theory hopefully should be a review with you guys, which is nice because a lot of times I have to give this talk and give like 2 minutes of here's what excitation-inhibition balance is. At least you guys have had at least an hour on it before. And then I'll talk about what I mean by structural heterogeneity. And this work was a collaboration with a group of anatomists in Germany. So I'll talk about the data that we got from them. And then talk about how the the relationship that we find between the structure, in particular heterogeneity of the structure, and how that impacts on excitation-inhibition balance. Some analytical work understanding the bounds of excitation-inhibition balance with regard to the structure, and finally a kind of fix to the problem that will arise along the way, using slow adaptation currents to correct for the structural heterogeneity. And stop me anytime you want for questions. Okay, so first of all, the background, which you heard about. So as we know, cortical spiking when measured is irregular and asynchronous. So any single neuron has roughly Poisson-like irregular firing with substantial CV of the ISI, and there are low correlations between neighboring neurons. In particular, I don't know if you guys have heard about it, or if you want to mention in the introduction, there's what are observed up and down states within cortical regions. In particular, the up states have this low synchrony in particular. And as Jonathan discussed, that property of cortical firing that was observed in the data was hard to explain theoretically for a while from simple models. And the reason it was hard to explain for simple models is because if you look at a single neuron that's within that network, and the ensemble of neurons around it are all firing irregularly and without synchrony, so then the net input that converges onto our single neuron should be approximately constant, basically just by a kind of law of large numbers argument. So we have some neuron that's summing k inputs, and if they're not correlated, then we expect that the sum should be fairly constant, and that the deviations from the mean, in particular the deviations for the mean, should be an order of magnitude smaller than the mean itself. Right? And that would lead to a constant input in time, and a constant input in time should yield, by any simple model, regular firing neurons, like clockwork. Right? Sorry? Okay, so the theoretical answer to that, which was proposed in the 90s and which you guys learned about, by Chaim and Carl van Vrieswijk in 1996, was that the irregular asynchronous firing could be maintained self-consistently if you have strong synapses and strong inhibition. In particular, inhibition that's strong enough to suppress runaway fire, and strong synapses in the sense originally meant, loosely speaking, strong synapses so that only a small fraction of presynaptic neurons have to fire to generate a spike. And the way that that's modeled, as you guys saw, the way that that's modeled is that each synapse is scaled as 1 over square root of k, where k is the average number of inputs. So the result of that is that the mean effectively is still larger than the fluctuations, but as you guys saw, the mean winds up canceling dynamically. So the excitatory current has an order of magnitude of square root k. And this is also similar to what we saw in the chaotic network, in the chaotic rate network, where we called it excitatory current. It wasn't really excitatory current in that context, but it was potentially excitatory current. It was potentially an order of magnitude of square root k. But we have strong inhibition, which will also provide a net current, which is order of magnitude of square root k. And what it turns out is that without any fine tuning, as long as inhibition is just strong enough, it's forced to dynamically balance, so that the rate of activity of the inhibitory neurons and the excitatory neurons are matched to exactly balance. And the result of that is that the residual fluctuations, which are small relative to each of these means, but after canceling the means, so the residual fluctuations, they become the dominant force in the network, and they actually drive the firing. Any questions? So here's what it looks like, as you guys saw. The single synapse within the network from population B onto population A, we've written JAB, where the JAB is order 1 parameter. So JAB divided by square root k will be a single synapse. And the result of that is that the net input onto neuron I in population A will have a square root k out front after summing over the k inputs. And then the sum, and here RE, RI, and RO are the mean firing rates of the excitatory, inhibitory, and an external population, which we just model as constant external population. Right, so that's
this is what I wrote as H external.
So that's the net input effectively onto every single neuron in the network. And the dynamic balance of excitation-inhibition requires that net input be order 1. And that yields the simple linear equations, which you guys saw on Thursday. So just two sets of linear equations for the two unknowns of the population rates. So given an external firing rate, an external drive, the two population rates will, it turns out, just match each other to solve this equation. And it turns out that will happen basically for every set of parameters in which such a solution exists with that allows positive firing rates. If positive firing rates can solve these two equations, it turns out that will be the fixed steady state of the network, and it's independent of the particular transfer function of the neuron. So integrate and fire was the context in which you've seen it, but largely, I guess I won't say too strongly that it's independent because there's certain, but it's not independent of all the properties of the single neuron, as we'll see by the end of the talk. But in particular, of the spike producing mechanism is independent of the particular features of that.
Specifically, excuse me for interrupting again. So one thing that is worthwhile noting, we discussed it in the framework of the integrated fire neuron, the threshold for spiking does not appear here. Like the important property of the neuron, taking these equations, this will not affect surprisingly, this will not really affect the firing of the neuron.
Yeah, and so you could have, there's all kinds of other models of neurons, quadratic integrate and fire, all kinds of other things. It turns out, so any of them yield these same rate equations. So the rate is kind of an emergent property of the network and not dependent on the particular single neuron transfer function in the balanced state.
Sorry, could you remind me what O is?
O is the external population.
What used to be H external.
H external and implementation. And this produces temporal irregularity as well as the low correlations, and it's managed to reproduce along the years a lot of experimental findings on the cortical firing properties. So it's a favorite model over the last bunch of years. But we wanted to ask, so all of that is in a context where the network structure is assumed to be uniform. There's essentially no structure in the network. The network is modeled as a random graph where each neuron is just connected to another with a fixed probability, which is uniform across the network.
Can I interrupt you because this is not exactly how I thought it. I thought a simpler version in which each neuron receives exactly the same number of inputs. And I just mentioned in the last minus 30 seconds that this can be generalized to the case that Itamar is discussing, which that we have a fixed probability of connection. In that case, you have some form of weak heterogeneity that would manifest as heterogeneity in the firing rate of the neurons, but this is not the heterogeneity that Itamar is talking about. He's talking about stronger heterogeneity.
Right. Yeah. So I'll describe what I mean. And the motivation was to think about, in the coming years, and already certain data is coming out, anatomical data about the connectivity within the cortex, and there's a major question about what are the impacts of having more realistic connectivity structures in the models which we study. So in particular, like I was saying, we study the heterogeneity of what we call the in degree. So the in degree is the number of inputs that every neuron gets. So in the model that Jonathan presented, it was exactly constant, Ki. The number of inputs to each neuron was just equal to K for every neuron. We'll look also at the relative in degree. It's just Ki divided by the average, divided by the mean across the population. So what I call homogeneous network, and these are the most commonly studied ones, is that where effectively the mean probability, the probability of connection is uniform. And so, like Jonathan just said, there's some amount of heterogeneity, but very small amount of heterogeneity, right? So when K is very large, every neuron has a probability of K over N of being connected to every other neuron. So it'll receive approximately k inputs plus or minus a square root of k correction term, so that the relative in degree will be right at 1 for all the neurons, plus or minus 1 over square root of k. So that's a small heterogeneity, and that's what we refer to as homogeneous networks. And this includes networks that can have different kinds of structure. They can have spatial structure, where the connectivity depends on distance, but still that the net number of inputs to each neuron is effectively constant over the population. And we wanted to know what would be the effect. First of all, what can we expect from real cortical networks? Can we expect that the number of inputs is really constant across the population? And secondly, what would be the impact if it's not constant? If there's significant heterogeneity, what would be the impact on the dynamics? And for example, if the network has heterogeneity to the extent that the relative in degree standard deviation could be just a fraction, 0.2. So people talk about in degree distributions in the wider world of networks and talk about power laws and long tails and all kinds of things. Here we're not talking about anything so complicated as that, just some non-zero significant heterogeneity, so that the standard deviation would be just a fraction of the mean. Yeah.
It is very important that all neurons in the network receive like a very similar number of connections, or it's just important like the balance per neuron, excitatory and inhibitory connections?
So that's a very good question. We'll see it. That's a great question, and we'll see it come up in equations later. Yeah. So how do we approach the question in terms of the biology? So we had this collaboration with Marcel Oberlander's group. He was in Tübingen and is now in Bonn. So they're a group of anatomists. They study the rat barrel cortex, and they've been one of their major projects was to construct what they call a statistical connectome. So these days people are working on electron microscopy connectomes, where you really open up a piece of cortex and really image the whole thing and reconstruct the whole thing in detail and pick out the connections between every single neuron in a real piece of tissue. Their approach is statistical, and what it means is that they reconstruct a larger piece of tissue, but not one whole piece of tissue. What they do effectively is go in and target single neurons, which are also able to record some activity from, and then stain them so that a single neuron within a piece of tissue has its whole axon and dendrite labeled, kind of like in this image here. And then afterwards do the slicing, and it allows the imaging to be a lot more efficient and fast. So they can do the imaging very, very quick and find the full structure of a single neuron. Now, the important thing that they're also able to do, which may be kind of somewhat special also to the barrel cortex. I won't go too much into the details of the barrel cortex, but it's the cortex, the sensory cortex of rodents, which is responding to primarily to the whiskers on the face. And it has this very nice organization which exactly matches the organization of the whiskers. So there's columns here in the upper left, real columns in the cortex, where each column has a primary whisker that activates it. And the columnar structure can be seen anatomically, primarily like the density of neurons, especially in layer 4, shows a columnar structure where there's a peak density in the center of the column, and then it goes down. So that means that there's a geometric kind of reference frame. So even though they're studying neurons in different animals, every time another rat, but they can register every neuron they find geometrically into what they call a common geometric reference frame. And they also, so additionally, they use genetic markers to label all the neurons in the whole region, and then have a map of the densities of neurons across the cortical region.
Reconstruction of single neurons using EM?
They use light microscopy. And they're able to do that because it's just a single neuron, so it's a sparse trace, and the imaging is fine and captures it, and it's awesome. So they don't.
What is the spatial resolution?
I don't remember. I don't remember in terms of the imaging itself. But what they do is they have the full soma density map as it were, right? After counting over a number of different individual animals, so they have the map of how dense the density changes within the cortical region. And then they have example neurons. So they have example neurons from different locations within that space. And what they do is an upscaling of the network, a constructing of a statistically representative, as it were, network, so that they, in silico, have this density of neurons. In our case, we're interested just in studying the layer 4 of a single column. So we start with a layer 4 column, which is about 3,600 neurons. And we have how the density is distributed in that column. So then the first step is to say, okay, let's randomly draw neuron locations, soma locations within that region. So then you sample about 3,600 locations within that space. Now each location has to get assigned a morphology, both an axonal and dendritic morphology. So they don't have the real axonal and dendritic morphologies, but they have a representative sample. I think they had, for example, like 30 some odd dendrites from layer 4 cells. So they draw from this library of dendrites randomly and assign to each soma in the location a dendrite and an axon. And then, so that's kind of a statistical representative upscaled network, full-size network. And then from there you need connectivity rules. And so there it's really kind of trying to use simplest assumptions without putting in too many assumptions. So they use a fairly simple assumption that the probability for connection really depends on there being some overlap between axon and dendrite. So two neurons have a probability of being connected if the axon of the presynaptic neuron has part of its branches that enter the space where the postsynaptic neuron has dendrites. So then it's just a matter of kind of calculating that kind of local overlap. So they break the space into voxels and calculate the local overlap of dendrites and axons to produce a probability of connection between each pair of neurons in this upscaled network.
How do the results depend on this probability?
So the results depend. The results of the full structure of this connectivity matrix that comes out depends a lot on that particular assumption. There's an assumption here of a kind of independence. An axon and dendrite, if they meet in one space, they'll have a probability of connection that's independent of whether they meet in other spaces, whether they have other connections, for example. That's one particular assumption that's been challenged in the literature. The results, as far as we're concerned, really don't depend on that, because at the end of the day, what we're studying is really just a simple second-order statistic of this matrix that comes out. Just that heterogeneity is really just the variance of the number of connections. So it doesn't depend too heavily on that. But
types of inhibitor neurons? Different types of inhibitor
different types.
What did you do there?
So they have different types of inhibitory neurons, but kind of only morphologically distinguished from one another. We kind of just lump them all together into one type. So that's the anatomical background in the work that they did. And what we get at the end is just a connectivity matrix. It's a full cell-to-cell connectivity matrix with a connection probability between every pair of cells in the network. And like I said, what we're interested in primarily is the heterogeneity of the in degree. So this is what we found in the connectivity matrix. On the left is the homogeneous network. So we construct that just by taking the mean probability of connection across the whole network and making a random network with E and I. So we constrain the mean probability of connection between each cell type. And then what you're seeing here is scatter plots. So each dot is a single neuron with the y-axis representing its excitatory in degree and the x-axis its inhibitory in degree. And you can see the matrix estimated from data has a substantial variance in the in degree. So the standard deviation of the relative in degree was 0.3, about a third of the mean, as opposed to what would happen if we had a random graph. So the data deviates significantly from a random graph. And the prime reasons for that are both the soma density is non-uniform within the space. So there's a bit thicker regions and less thick regions where there are more cells. And the morphologies are very heterogeneous. So every given cell has a different sized dendrite, different sized axon, sending its branches to different locations. It's going to receive a different amount of inputs. So that's the first observation, empirical-ish observation. And then the second one is that you can see that there's a clear correlation between the excitatory and inhibitory degrees that shows up on the scatter plot. So a neuron that gets more excitatory inputs also tends to get more inhibitory inputs, and so that gets to the question that was asked before. And so we'll see kind of the feeling might be that correlation could compensate for the heterogeneity. We'll get into that coming up. Okay, so first of all, what is the impact of the observed heterogeneity on the dynamics? So first of all, just straight into simulations of integrate and fire neurons of the whole network, and comparing again the random graph network where the structure is uniform and the in degree is homogeneous to the network that came from the estimates of the anatomy. And you can see right away, matching the mean firing rates across the population, we get very different results in terms of actual dynamics of the population. So this is a raster plots. Here we're seeing 500 milliseconds of time and neurons on the y-axis. So you can see on the left, the homogeneous network has this kind of classic balance type irregular firing, what we expect from cortical neurons to look like, that the firing is asynchronous and irregular and spread across the population. Now in our matrix from data, the anatomically constrained matrix, we still have asynchronous firing, but we see that only a very, very small fraction of neurons is active, and they appear to be active at very high rates. Most of the neurons are completely silent. If we look at the rate distribution, the distribution of firing rates across the population, so the homogeneous network gives us, as expected, a realistic population firing rate distribution, which is roughly log normal, which has been observed in experiments, and almost every single neuron is active. When we look at the matrix, the anatomically constrained matrix from data, we see here this bar is actually fully zeros, totally silent for a 60-second simulation. That's over more than 70% of the neurons are totally silent, and the maximal firing rates go up well above 100 hertz, which is highly unusual.
The thing that the firing rate, the distribution of firing rates is the log normal. This is first, as you said, something that you observe in cortical networks, and second, this is a signature of balanced networks framework. This is not it's not the result of a particular parameters used by Itamar. It's actually pretty interesting why you see it.
What does it mean?
I'm sorry?
What does it mean, that it's log normal?
Well, it means that if you look at the distribution of, take all the neurons in the network, each one you measure the firing rate, you take the logarithm of this firing rate, and you plot a histogram, you get a normal distribution.
So that's these insets are the log distributions of the firing rates, and so it's roughly log normal.
And this is also what you see in the experiments. And we now understand why log normal distribution of firing rates is something that naturally emerges in homogeneous balanced networks. So what I did in class was to assume, in 2 minutes, what I did in class was to assume that all neurons receive exactly the same number of inputs, and the same number of connections. In the homogeneous case, what you assume that statistics receive the same, the probability of connection is equal for all neurons, and as a result, some neurons receive slightly more input and some neurons receive slightly less input. Because they receive input from a large number of neurons, then if you look at the distribution of input to the neurons in the network, this distribution is what? How should the distribution look like? So the number of inputs per neuron?
the large N limit.
Larger N. Normal. So the distribution of number of inputs per neuron is normal. Now the question is, why is if the distribution of inputs is normal, why is the distribution of firing rate log normal? And to understand this, the way to think about it is that each neuron receives average inputs and fluctuations, temporal fluctuations on top of this average input. Those that receive a stronger input, those that receive more inputs will receive on average larger DC. Those that receive a smaller number of inputs will, on average, the average input, well, trivial, will be lower. So those that receive more input, on average, will be closer to the threshold. Those that receive less input will be further away from the threshold. Now, so you have neurons that, on average, are further away or closer to the threshold. Now, it turns out that the dependence of the firing rate on the distance from the threshold is exponential. For the physicists among you, the reason that it's exponential, this is like a particle in a double well potential. The transition probability depends on the energy gap. So we have something similar here, that the probability of crossing the threshold is exponential in the minus the distance, and this distance is determined by the average number of inputs. So in fact, the fact that these neurons are firing Poisson-like, and the fact that the distribution of firing rates across population is log normal, these two properties come from they have a common mechanism, which is pretty interesting.
Yeah. And that's work David Hansel has published not that long ago.
2011, I think.
Yeah. And so that's in the standard balanced state network with homogeneous connectivity. As I said, in our network, we see a very skewed firing rate distribution, where over 70% of the neurons were totally silent. Also, if we look at the irregularity, so the homogeneous network gives us Poisson-like firing, where the CV of the ISI is around 1 or even higher, and even grows with the firing rate. Our anatomically constrained network, coming from the estimates from data, have more regular firing. And with increased network firing rate, the regularity increases, so the CV drops significantly. So that small fraction of neurons that fire, they fire at high rates and also more regularly. And finally, we also know that we expect the linear relationship given by the balanced state theory. So the homogeneous network nicely reproduces the predicted population average linear rates, but the anatomically constrained network deviates significantly from what we would predict, just based on the balanced theory.
But this is not something that you measure experimentally, right?
I mean, this is a simulation result.
Unlike the distribution of firing rates, the
right.
distribution of firing rates is something that you care about because this is something that you measure
right.
with experiment. The CV is something that you care about because you measure it experimentally. Yeah. So you're unhappy when your theory is inconsistent with measurement. But this is something that
right. This is something that more the theory cares about, right? So this is an element that says the theory is wrong, not just quantitatively in what we saw by the firing rate distribution and the CV-ISI, but on a more fundamental level in terms of the theory. This says the theory is wrong, because the first thing the theory tells us before the CV exactly, also quantifying the CV from the theory itself is very difficult, and the firing rate distribution. So the very first thing the theory tells us is that the firing rates should match these predictions. So you're right. In terms of the biology, the first two things are the more important, the more interesting things.
The theory, we want to understand the brain.
Right. We want the theory to be right, though. So this is kind of the knockdown to the theory. So the theory could have been right, and then we could have had a different distribution, firing rate distribution. So we would have had to study more in detail. The CV of the ISI wasn't 0; it was just smaller than 1. So you could have a balanced state with smaller CV. You could have a firing rate balanced state with broader distribution of
So I
firing
want to
rates.
understand your
My point is this breaks the balance theory for sure.
This means that the previous equations are
Yeah.
previous analytical equations are inconsistent with the simulation.
Yeah, exactly.
But what you showed us previously tells us that simulations are inconsistent with the brain.
Right.
Which is, I find more, I'm more concerned about
Right.
the scientists who cared about the brain.
Great. Yeah, I think this is then the step to then understanding that. So why were our simulations not matching the observed biological reality? And our way to access that question is through the theory. What does the theory tell us about how the simulations are behaving? So first of all, the theory tells us that the simulations are not behaving according to the theory. So then we can try to understand what the theory, a corrected theory, would say about that, and from that come back to say, okay, why was that? Why were those same simulations producing results that don't match biological reality? Okay, so to try to understand from the theoretical side, let's go back to those same balanced equations. So we had the net input onto a typical neuron, and from that we derived those two equations, which the simulations didn't match. Now, we also saw this picture before of the net excitatory current and net inhibitory current, and talked about the dynamic balance that's represented by the equations we just had up here. Now, in the original homogeneous network, so that single trace might represent a single neuron, and the dotted line, though, could represent the time average, and it would be roughly identical for all the neurons in the network. So we could write a single equation, really, that represented all of the population, the whole population. Typical neuron has inhibitory input right around here with some kind of small deviations from that. And from that we could derive the equations that were relevant for the whole population. But now in the heterogeneous network we simply have a broad distribution of inputs, so that we have a really broad distribution, a broad distribution of the in degrees yields a very broad distribution across the population of the net input. So these kind of schematic drawings would represent the distribution across the populations of the net excitatory, inhibitory, and external inputs. And if we sum those three together, naively at least, what we expect is that the net input no longer settles right around the threshold, but the net input itself just simply has a broad distribution across the population, where a lot of neurons have their net input very far below the threshold, and we're talking about, in terms of the model, an order of square root of k below, so a very large deviation well below the threshold, and so that explains the neurons that never fired in our simulations. And we could have a tail that reaches up well above threshold in the positive direction, which would explain the high firing neurons, as well as the constantly firing, the regular firing neurons that have their average input way above threshold are expected to fire regularly. So that's naively a pretty straightforward and simple explanation. We have a lot of heterogeneity structurally. That would yield heterogeneity in the net input, and that would stretch the population, break the balance in the sense of stretching the population distribution. One question, again, back to the question that was already asked, though, what about correlations? So really, as we saw in the data, we have a situation where neurons that get more excitation tend to also get more inhibition. So potentially, if we summed these three distributions, but they were highly correlated, and that each cell's excitatory input was predicted from its inhibitory input, we could get a situation maybe where the net current would actually still remain near the threshold. So it's not necessarily clear why we don't see that in the simulations. Let's just go and see basically what I said now in terms of the equations. So these were the two equations before. Now effectively what we have to do, we can't represent the whole network by a typical current that represents the whole population. We have to write really a different net input for every given cell, which depends on the number of excitatory, inhibitory, and external inputs that cell receives. And that gives us a whole set of equations, not just two equations representing the whole population, but in principle a different equation for every single cell. And these equations can't be solved. So algebraically, basically the algebraic flip side of what we saw, kind of geometrically what we saw in that picture, is that this set of equations here now representing every single neuron's local balance, the only time it can be really solved in the sense of the excitatory and inhibitory firing rates doing what they need to do to cancel the net input, is if it just so happens that the excitatory, inhibitory, and external inputs are all perfectly correlated. So if they are perfectly correlated, if KIE equals KII equals KIO, then we could just pull that term, the common term, out, and we would have balance again. So it's in principle possible for a network with a broad distribution of indegrees to have balance, but it would require that the excitatory, inhibitory, and external indegrees, the three different sets of indegrees, would have to all be aligned. And now we wanted to also quantify that.
This point clear? That one is crucial.
Yeah.
And previously we had two equations with two unknowns. Now we have n equations with two unknowns. And in general, they cannot be classified.
Okay. So we wanted to quantify this issue of the correlations. To what extent can correlations recover and facilitate balance? And why don't we see the correlations which we did observe in the data? Why isn't that enough? So we have a simple model of a network with broad indegrees and correlations. So we can build a generative model, build a network where we give each neuron, we assign each neuron its indegree, and we take that indegree from some distribution. So we can take it from a distribution with a given variance, and also with a given correlation, so that we choose with a parameter C how strongly correlated the number of inhibitory and number of excitatory inputs will be onto each neuron. So if C is 1, we have a perfectly correlated population, which can still have significant heterogeneity in the indegree. And like we saw before, what we would want is for every single neuron's input to be balanced. It turns out, without going through the details here, that can be calculated in a pretty straightforward way. For this model, the requirements on these two parameters, the variance of effectively the heterogeneity of the indegree and the correlations between different populations, essentially their product has to be very, very small. So it turns out sigma squared times 1 minus c has to be on the order of magnitude of 1 over k. If that is satisfied, then we'll have balance on that individual neuron level. Every single neuron's net input will be near threshold. But it's a pretty tight bound. So if sigma, we saw the case where sigma was 0.3, for example, coming from the data. So when sigma equals 0.3, we require a correlation, which is like something like 95% correlated, 0.9 something, I don't remember exactly, 0.93, or something like that. Correlations that are very close to 1. So what we saw originally in the data is that there is correlation, but it's nowhere near that strong. It's something like 0.5 to 0.7, maybe even 0.8 correlations between excitatory and inhibitory indegree, and that's not anywhere near the 1 over k bound. And to test that theory and kind of further justify that point, we ran simulations of that constructed network model with ranging those two parameters, so ranging the heterogeneity of the indegree and the extent of correlations between E and I and external. And what we see here is the CV of the ISI for every single point. So every point in space is a different simulation of a different network with different structural parameters, and the color represents the CV of the ISI. So the yellow is the CV is up around 1, so as we expect. So networks with very low heterogeneity, that's going back to the homogeneous case, as we expect to have CV around 1. And further, we can see that, in fact, indeed, we do see on the right-hand side, going up the y-axis, we're increasing the heterogeneity of the indegree, but with high correlations, and there we are able to maintain the high CV. But networks without really high correlations very quickly go to low CVs.
In this figure, where is the anatomical data?
I'll get to that in just a second. Good question. We did the same thing with just measuring the fraction of neurons that were totally silent over a 60-second simulation. So we can see we have this separation, this kind of crossover between a balanced regime in which really almost every single neuron in the population is active as we expect, everything's engaged, to a crossover to a regime where up to 0.9, between 0.7 and 0.9 of the population are totally silent. And the black line is exactly that equation, that inequality that I wrote, sigma squared times 1 minus c is smaller than 1 over k. That's the black line, which fits fairly nicely. So the fit is fairly nice. And here, to Jonathan's question, this is the area where the correlations from the data were found. Again, there's different correlations because there's correlation between E and I, correlation between I and the external, correlation between E and the external. They would all have to be aligned in order to fit the.
Data on external?
Yes, we do have data on the external. I didn't mention it. Yeah, they have axons coming in from the thalamus as well. Yeah.
Still you can.
And yeah, so that explains nicely. So we have significant correlations which do mitigate the effect of the structural heterogeneity, but still leave us outside of the regime where we would expect balance to be held, and that's what we observed in the simulations. And that's our explanation for why these simulations, when we just take the anatomical data right off the shelf and try to run simulations, the biological things, properties that we expect, the high CV and the nice log normal firing rate distributions don't emerge, and that's a result of kind of this deviation from the balanced regime in terms of the structure. Okay, I have one more section, and we'll take a break. Okay, so we'll take a break and then come back to, all right, so that's a problem. The biologically inspired connectivity matrix doesn't replicate biological firing properties. So what kind of fixes do we need to make to our model to recover the predicted kinds of dynamics? Come back at 1:05. בפורום, מישהו שאל את זה בפורום. נכון. טוב, אז אולי זה לא יתפר. אני ממשיכו. אז זה בהחלט הסיבה שזה יכול ברגע שיש לך דינמית יותר ארוך, אז יש לך יותר אינדיק. טוב, זה יותר תקליט. אז אולי משפיעים, ואז זה לגבץ את הנקודה. זה נכון, זה אנחנו תלוי בדברים אחרים, אבל כן, זה נכון. העניין הוא שיש הידידות לחודקים סינפטיים, זה ממש לא ברור שזה באמת תלוי. בעצם ההצנות בין הבייזל סינפטיים הקרובים לאפטיקל, זה לא ברור כי שיש תמיד פיזור אחרים בין האקספט
שזה משפיע.
לא, כאילו קומפלמנטרי.
כאילו,
יש לגבי מודלים עוד יותר ביולוגיים ולחשוב על למה זה מאוד לכולם, מי שמבין בזה לחשוב על זה, יש קצת בעייתים, זה דבר אחד,
שניהם לא אמורים איזה אחד
אני חושב שאיכותית, אני לא חושב שזה
כן, מה נראה שיהיה?
יום ראשון וחמישי אם הם יעלו
תהיה זה המרחק בין
לא,
כן יש תיקונים שכבר הראיתי כן, תגיד לנו מרי אבל לא מה שיש ויש לי, כן, אני עולה תכף את
שאלה בנוגע לתרגיל, יכול להיות שהיה לכם שם טעות בניסוח, כתבת פה לסקשן מסוים לעשות DT נורא קטן, אמרת שזה 0.001 טאו, נכון? לא התכוונת 0.001, זה יותר גדול מטאו.
לא,
מה?
מילי שנייה, זה כן, טאו זה מילי שנייה?
טאו זה מילי שנייה, ואז אמרת תעשו DT שהוא 0.001, שזה...
מילי שנייה.
אז זה בגודל של טאו. בעצם אתה אומר לעשות את ה...
0.01 מילי שנייה, כלומר...
0.01 כפול טאו. כן, זה מה שחשבתי.
שזה אותו דבר.
לא, זה לא כפול.
לא, נקודה 0.01 במילי שניות.
במילי שניות, בדיוק, בטאו. כאילו אם אתה עושה בסקט, אז זה נקודה 1 טאו.
זה באמת כמה זמן? לא, כי טאו זה מילי שנייה.
כי טאו הוא מילי שנייה. והוא התכוון ליחידות של מילי שנייה. כי
אחרי
אחרי זה יותר גדול, טיימסטייפ יותר גדול מבדרך מהרגיל.
כמה זמן זה לקח?
לוקח המון זמן. מתחילת ההפסקה אני לא
לא, זה הגיע
50.
עוד יש תחת לא
זה לא חזק, הוא לא חזק.
הוא יותר חלק הממוצע
אני רק שזה לקח
למה? יש מערכת
אם זה לקח דקה או שתי
אז אני לא מבינה מה הבעיה,
אולי יש עוד משהו אחר שם
מה אתה רוצה לבחור בינתיים?
אולי יש עוד משהו
מה האורך של הסימפטים? למה צריכה להגיע ל-50,000?
כי יהיה 50,000 כאילו צעדים
אחרי שאתה מגיע לסוף, אחרי שאתה מחשב את כל הימים, כאילו...
50,000
בסוף לא ענית לי בפורום לגבי מה צריכים לעשות עם הטיימסקייל, אורדר פלאגין מספיק או לא? זהו,
סוף כזה?
התחילה בו את הישב לא יודע, זה לא מעניינים אותי בבעיה כן, זה יתכנס נכון.
כן, בסוף.
כלומר, x, y כתוב x.
בפיזיקה במיוחד, יש כל כך הרבה אפשרויות. אחרי, כן, כל מיני כיוונים.
גם לא להמשיך לאקדמיה, אלא להמשיך לתעשייה, אבל גם אפשרות.
גם אחרי זה, אתה לא נפתח, גם בדוקטורט בפיזיקה, הרבה אנשים בספורט, ממשיכים למשהו אחר לגמרי.
לא הגיע לגודל, פעם לגבי... לא משנה, אבל הבנת את הרעיון. פה אתה בוחר את איזה סוג של התאמה אתה רוצה לעשות, את הניהול, את היותר ראשונה, למשל, זה נותן לך את ההתאמה. ראשונה. אז אחרי שתעבירי זה לא. ואז זה נותן לך את ההתאמה. בפרט פרט ג'נרייט קוד, לא יכול לעשות את לפני, שאת מכניסה את תכתבי לוג של ההתאמה של זה. ואז כשאת נמצאת פה, את יכולה כשאת נמצאת תפקיד שאת עושה, או בעצם פרופייל, ג'נרייט קוד, ואז הוא מייצר לך פונקציה שקוראים לה ERC, שפה את מכניס את הפיד שלך, או שזה לא יהיה, פה כרגע הוא לא עשה אפילו כשאת תעשי את הפיד כמו שצריך, הוא לוקח את הפנימה, את ה-X ואת ה-Y שאת עושה, הוא מייצר את הפונקציה הזאת, ופה הוא נותן לך חזרה את התוצאות של הפיד, שיש כזה, ואת כמה הוא צורך. אז מפה את יכולה למצוא את המידה, את יכולה להשתמש בפונקציה הזאת, כדי לעשות פיד שבא לך. פשוט תעשי את הפיד. זהו, אולי בוא נעשה. בוא נעשה. פיד שווה...
אתה בטח לא
אז 100 ו-y שווה... אוקיי, עכשיו אנחנו צריכים להוסיף.
מה שאתה תפריד לי, יש לי כאילו יוצא לי שאין
וואי, הקו הליניארי הזה שעשיתי, נכון? אתה כבר רצה לו פיד, נכון? כבר יש לי כבר יש לי פיד, זה כותב לך שהפיד מושלם כמובן. יש פה את ה-Deviate. P1 זה פשוט הקו הזה ו-P2 זה החיתוך עם צירים, נכון? כי זה הפונקציה. יש לנו את P1X ועוד P2. כן, אבל הוא הכי אפס שנעשה בזה. יש לו, תלוי, זה לפעמים יש לו שגיאות נומריות קלות. אבל זה בסוף פיד. יופי, אז זה אותו פיד, אז זה נותן לנו את הפיד הפולינומי. בסדר? עכשיו אני אצא נגיד, אני רוצה שעבור כל מיני דאטה, לאו דווקא בשביל y יהיה לי את הפונקציה של הפיד הזה, אז אני אעשה לו פייל, generate code, הפיד ספציפי הזה שעכשיו עשיתי, עכשיו את רואה, כבר בגלל שהכנסתי לו. אז יש לי straight feed ty, כלומר הוא לוקח את הערך של t, הוא לוקח את הערך של y, ומוציא לי את הפיד ריזולט. אז אם עכשיו אני אשתמש בפונקציה הזאת, אני אשמר אותה. טוב, נקרא לה בו. חוזר אליי נגיד, עכשיו אני יכול לכתוב לו, פה אני יכול לכתוב לו, לא יודע, A שווה create feed, גדול, create feed, ty, מוציא לי את הגרף, אני יכול להגיד לו לעשות את זה, שפה מוציא לי את הגרף עם הפיד ביחד, והוא נותן לי את A הזה שזה איזה object, שאם אני נוגד אז כותב מה יצא לי A.P1, הוא נותן לי את ההתאמה A.P2, נותן לי את כן, את השיפוע, A.P1 נותן לי את השיפוע. ואז אני יכול להכניס פה איזושהי פונקציה שאני רוצה. עכשיו במקום T ואת Y אני יכול להכניס פה את ה-DS שלה, את הלוג שלה, כאילו לוג מה שלא יש. תמיד ייתן את הפיד הפולינומי, הוא לקח בדיוק את הפרמטרים שהשתמשתי בהם פה, הוא ייקח בדיוק את הפרמטרים שהשתמשתי בהם, שביקשתי ממנו. כל פעם אני יכול לעשות פיד אחר וליצור פונקציה חדשה. הדבר הכי
בדיוק. כאילו, הוא אתה רוצה שבאמת הוא יישאר ועובד,
נראה לי הוא מסיים את החלק האחרון, לראות מה הוא עשה, עד עכשיו הוא עשה נראה לי הקדמות למה שהוא עשה.
בא לי להגיד שסתם המקצוע זה לא משנה. הסדר של המקצוע לא משנה. כשאתה בודק את השונות של זה, זה משתנה לאורך
זה לא באמת צודק.
אם אפשר להגיש אותו אחרי סוף תקופת
אפשר להגיש אותו עוד שנה וחצי. הוא יגיע לסדר. לא, זה קשה.
Okay, so let's continue. We've introduced and understood the problem, that structural heterogeneity in terms of the number of inputs across a population within a network can prevent excitation-inhibition balance and lead to a state where only a small fraction of neurons are active at very high rates and with regular firing properties. And now we're going to introduce a potential solution to this problem. So neurons are observed to have adaptation currents. So the direct observation, and is most commonly known for excitatory neurons in the cortex, the direct observation is if you drive a neuron with a current into the cell and watch it fire, the firing rate will not just be a constant function of the strength of the input current. It'll decay over time. So it has a kind of decay, which is interpreted but also then further studied mechanistically as what's called an adaptation current. Those mechanistically, people talk about sodium-dependent and calcium-dependent potassium channels. So they have slower time scales, much slower time scales than definitely than the spike, also much slower than just the refractory period. And the time scales that are observed for excitatory neurons are on the order of 300 to 500 milliseconds, after which there's a noticeable decay, for example, in the firing rate. It turns out, so that's well known, is that excitatory neurons have these adaptation currents. What's less well known and less well studied is that inhibitory neurons also have adaptation currents. Some classes of inhibitory neurons have been shown clearly to have somewhat roughly similar adaptation properties as to the excitatory neurons. But other inhibitory neurons are called regular firing cells. That's one of the names they're called because they're thought to not have this adaptation property. But it turns out a few studies have studied these neurons at longer time scales, something like on the order of 20 seconds. So in those studies, people gave sine wave currents to neurons over longer time periods and count the number of spikes on the first up cycle, on the second up cycle, on the third up cycle, and so on, and found that even these inhibitory regular firing, fast spiking neurons do have measurable, noticeable adaptation over those longer time scales. So we introduced to our model integrating firing neurons an adaptation current. So this is the form of the adaptation current we introduced. So the current I adaptation, this is going to be introduced, it's going to be added to the cell's current, subtracted from the cell's current, and it's going to come in as a negative current. And the way that it works, the way this model, and this is a fairly standard model for adaptation, it's probably one of the simplest forms of it, what's called spike frequency adaptation. So we have it over here, a delta function with TSP. TSP is the time of a spike. So every neuron, when it fires a spike, in our model, so when it fires a spike, this adaptation current, the negative current, is increased by some amount, and we model it as a relatively small amount. It's about the size, order of magnitude of a single synapse. So when a cell fires a spike, it has an internally generated negative current in our model, and so the size, the amplitude of that current after it spikes is here J adaptation divided by square root K in our scaling, and the time scale is given by the left side over here of this equation, right? So what we'll see here is a dynamic equation of the adaptation current, and if we solve it, we'll get basically just an exponential decay, right? After every spike, we'll have a pulse up, and then an exponential decay, and the time scale of exponential k we model as k tau. So tau being on the order of magnitude of the synapses, about a millisecond, 2 milliseconds, and k as the number of connections. So this gives us in our model and in our simulations adaptation time that we modeled. We made it dependent on the cell type. So we came out with a model that had an adaptation time scale for excitatory neurons of about 500 milliseconds, and for the inhibitory neurons about 2 seconds, which was found in some of those papers as the average adaptation time. So that's the model, and that's every single neuron after it spikes has a negative current, which is not so big on its own and decays very slowly over time. Question?
Yeah. Slices. So you take a slice, you inject current to the neuron. So this model predicts that the time rate, time scales of a second will be almost independent of the input. Right?
If you put in a fixed drive, you're saying?
So adaptation has to be finely tuned up to square root, one of the square root of k.
Not in our model. In a slice where you deconnect, take it out.
No,
No, but you'll see what it does in our model. If you're asking about a slice where there's no network,
I'm looking at these equations.
it doesn't have to be fine-tuned. That's the order of magnitude. Maybe I'll go forward with the analysis. So what will come of this is if we have a neuron that itself is firing at some firing rate r, firing irregularly, so then the time averaged, that adaptation current, its average over time will be square root of k, right? Basically the long time scale, which has a factor of k in it, time multiplied by the relatively small single amplitude of the pulse gives us the average, or the area under the curve, which will give us a square root k times these order of 1 parameters. And so that's the motivation for scaling it this way. So now we have this adaptation current that will enter the leading term of our net input. So this term will be subtracted from the individual cell's current and will be dependent on that individual cell's firing rate. So let's turn to that net input. So we have, back to the same equations that we've seen before, the net input of a single cell, which depends on its in degree, and the firing rates of the excitatory and inhibitory populations. And now we've subtracted this term that depends on that own single cell's firing rate. So that having the own single cell firing rate gives us this expansion of the degrees of freedom algebraically, where what we had previously was a set of n equations with 2 unknowns. Now effectively by giving the cell its adaptation, it's this strong dependence on its own firing, so we have now n unknowns entering our equations. Every single cell's firing rate individually can influence its net input. And now we want balance to happen locally. That means that we want this term, the net term, to be order 1, or inside the parentheses, we need that to effectively be small. In the large k limit, we need that to be 0. So we can look at that term inside the parentheses and solve for the balance condition, that gives us a local balance equation for each cell, and it can be solved very easily for each cell's firing rate. So just the parentheses, setting that parentheses equal to 0, and then dividing by j to n tau gives us this equation, which is it gives us the necessary firing rate for each cell. Each cell that has net positive drive will have to match, will have to balance its own net drive from the network by matching this firing rate to this local balance equation. And from there we can go back. Now this still depends on the firing rate. This expression depends on the firing rate of the population. So to have a full solution and a self-consistent solution, we now also have to average over the single cell firing rates on the left-hand side, and that'll give us an average rate R for each population, and then that has to be the same as what we wrote on the right-hand side. So solving that in a particular regime, so in a particular regime, basically if the adaptation terms here are strong enough, then it turns out all of these will be positive. All of the cells will receive net positive input, and in that case, the expression is totally linear. And we can average naively on the left-hand side and just get exactly RA, and then kind of rearranging terms, we would cover balance equations that look very similar to the classic two balance equations. Basically, we just wind up subtracting the effective adaptation current from the excitatory drive, and also similarly for the inhibitory population, subtracting the net adaptation from its inhibitory drive. And there's the two balance equations, global balance equations for the two unknowns. And so that's our theory. That's our model.
I'm going back to my question
Yeah.
about slicing. So you have the predictions about the response of neurons to current injection, and it seems to me that the adaptation that you predict is much stronger than the one actually observed.
I mean, the parameters that we wind up using here were fit to experimental measures of adaptation in terms of the time scale and in terms of the amplitude. Yeah. It's a question of how big this J ad is. It's order one, so we took to be a fraction, to a fraction of 1. Yeah. Right. So it's true. This was one of the reviewer's major concerns. Is this realistic? And so we had a literature search that showed that it's within the order of magnitude. We wound up having to use a stronger adaptation current than is found in an experiment to fully recover the full population activity. So kind of effectively, if we scale things as order square root k to match the findings of the adaptation strengths, we need this J adaptation to be like 0.2 or something like that, kind of small order 1 to be realistic or something like that. But it turns out that's enough to have a substantial effect on the dynamics. Maybe not fully enough to recover all the cells firing, but it has definitely a substantial impact already. This here is without regard for the biological reality of those parameters, just a check of the theory. Does the theory work in simulations where we ignore biological reality? And indeed it does. So these are the population rates as a function of the external drive, recovering that linear form from the original balance scenario, but now with that tweak with the adaptation. And then we can look at the raster plots to see just all the neurons are firing. The firing structure is irregular and asynchronous. And we can see that those measures that we looked at before that were of interest biologically, the CV of the ISI and the fraction of neurons that are silent. So here in black is this adaptation, this network with adaptation, and in blue is a comparable network without adaptation. And we can see that the network with slow adaptation has irregular firing, and all the neurons are active. So there's a couple interesting things here I think about the result. One thing, we're adding this term that has this kind of slow negative feedback, which naively one would expect kind of creates a kind of regularity into the dynamics. It's been shown in other places that adaptation currents can drive oscillations, can actually make neurons fully oscillatory. And here we introduce that kind of term, and it actually increases the irregularity. So it's a little bit counterintuitive, but it does that by canceling the mean dominant part of the input, canceling it, and then allowing the fluctuations to dominate. It kind of frees the fluctuations to be dominant again. And then the other thing, which is also somewhat not immediately intuitive, is so we thought the adaptation works. It's clear that a neuron which has net positive input and is being driven very fast, the adaptation will drive down its net input and kind of bring back its firing rates to more realistic firing rates. But what we see here is that also those neurons which otherwise would have had negative input and been very low beyond the threshold and not fire at all, in theory, kind of intuitively, adaptation is not acting on them at all, because if they're not firing, there's no adaptation current. But it turns out in the net kind of collaborative network effect, the adaptation current allows those neurons, winds up in the balance, bringing up their net input so that they're also able to fire and have their net input be close to the threshold. So that's kind of an interesting emergent property of the interaction between that individual cell property of the adaptation current interacting with the population balance. And then just finally, those individual rates, that local individual balance equation that I wrote also in simulation predicts very well the local individual firing rates are well predicted by the theory. Finally, we went to the more biologically plausible realm of parameters and back to the anatomical connectivity matrix and to an adaptation currents which are at least close to and within an order of magnitude of what's been observed experimentally to see if that kind of range of parameters would bring back regular typical kinds of cortical firing properties. Particularly, we simulated this somewhat old by now experimental paradigm where a whisker is kind of offset for 200 milliseconds. It's called a ramp and hold stimulus, which it's not so popular anymore, but it served our purposes. So we simulated that kind of an input, and also spontaneous firing. So we found in the spontaneous firing, when we put in the biologically plausible parameters of adaptation that we found in the literature, we find a return to realistic firing rates where all the neurons are active in spontaneous. The firing rates, at least much closer to log normal. It's not exactly log normal. And in the stimulus evoked phase also, we found kind of that all the neurons respond to the stimulus, which was not the case before adding adaptation. So that's basically the end of the story, at least that I'll tell here. The summary, so the anatomical data that we received estimating the connectivity structure predicts heterogeneous in degrees in even local cortical networks, such that the width of the in-degree distribution is a significant fraction of the mean. Because of that heterogeneity, E&I balance would be expected to be prevented, and the resulting state would be exceedingly sparse with only a small fraction of neurons firing at high rates and low regularity. Correlations between E and I in degrees can mitigate the impact of the heterogeneity, but they're not enough according to our theory, and the simulation results are not enough to really recover balance and fluctuation-driven dynamics. And we showed that the slow adaptation currents, at least close to biologically plausible parameters, can facilitate this global balance with a novel dynamics with an interaction between this local neuron single cell property and the global balance. That's it. That's the story. One reviewer wasn't satisfied and didn't like the adaptation and asked about plasticity. Couldn't you just fix the strength of the synapses? Which makes sense. We don't object to that. What about the strength of the synapses? Here we just assumed all the synapses have the same strength, so a cell that has more synapses also has stronger net input, but maybe there's plasticity at work, so we went back and also alternatively introduced a plasticity rule that can recover balance by adjusting the weights. That turned out to be relatively simple to do, so that's also in the paper, but I think less interesting. That's it.
Why do you think
Less interesting from the beauty of the theory side. Biologically, it's definitely it's.
And people have shown.
Yeah, biologically, it's interesting and expected. Really, it is kind of the more naive thing that you'd expect. Okay, if you have a non-biological structure, so there's lots of ways that homeostatic mechanisms can fix it. And more likely, yeah, and this is a known one, so less interesting in the sense it's less novel and.
Less novel given the first one.
Yeah.
Given that you start.
Yeah, that's it. And I just wanted to also mention another LSEC outstanding publication, which was given out on Thursday, which also happens to be about excitation-inhibition balance by Ron Darshan and David Hansel, published in Nature Communications just like a month ago. A really great paper. You should read it. That's it. Thanks. Timescale, right. רוצים שאני דקה על timescale.
Timescale.
Yeah. Just a second. Okay, so in the exercise, I'll leave this for you.
Yeah.
You can plug in the meantime. Okay. Okay, so we have the exercise with the Markov chain, and asked you to say what the time scale is of arriving at the steady state without defining in the question what we mean by time scale. So the hint that I would give is when we talked about time scales in the past, we talk about time scale in particular when we have some kind of exponential process in the time scale, and when we have an exponential, right, we have e to the minus lambda t, so the time scale would be what? 1 over lambda. Okay, so that's basically my hint. I kind of feel like I want to almost just stop right there and just say that e to the minus t over tau, right? If you have e to the minus t over tau, tau is your time scale, and so write that, plug that in to right. So you have that's the hint. So you have a process that has lambda to the n in terms of time steps. If you write that as an exponent, then you can pull out that tau, write it as e to the minus t over tau, and extract tau.
Okay, we have 20 minutes to summarize the course. So just copying the slides from my first presentation. One thing that you may find of interest is the fact that there is an exam. It's an open notebooks exam. Basically, you can bring your notes. Grade would be 70% exam, 30% homework assignments. Video recording, as predicted, it was not 100%. One thing that I wanted to discuss with you is the extra hour. I kind of felt that it was not... well, did you find it useful, the one that used it? Yes.
It was useful. It was most useful when we worked with Itamar about things we didn't do yet in class.
You did not do.
When it's
predictive hour.
when it was a predictive hour about theoretical background that you used.
So rather than answering questions about things that you don't know, it was answering questions about things that you don't know that you don't know.
Give us mathematical background to help us be more alert in class with you afterwards.
Which is, yeah, okay. It's more challenging for us because it's not always clear what are the things that you don't know. Yes?
I think that, when we come with questions, it's kind of office hour. And I think people that want to ask questions usually go to office hours, so it's better to have something more formal, as Dino said, or maybe to learn something before class or after class, or
What do you mean by more
type of
formal?
structure. So it won't be just spontaneous according to our questions, but something more structured.
I'm sorry? Okay. But you still think that it's useful? Yes?
Maybe just the fact that we didn't know about it right until the semester began, the people who are not elder students, we couldn't keep up with our hours and other courses. So if we would know about it before, we might have the chance to get to these hours.
Itamar, this is important, we should discuss. Well, everything is important, but this is easily solvable. So maybe this is something to discuss with Neely. So add it to the Shnaton. Okay. There was one thing that was introduced by Itamar against my better judgment this year, the fact that we had 2 hours and 2 hours rather than 4 hours on Thursday. So do you support Itamar or do you support me? Okay, you will pay for this lack of loyalty. Okay. Okay, so you think that we should keep the extra hour but make it perhaps more prepared and okay. Are there specific things that you would have liked Itamar to do before, so that you can tell us next year do this, teach this before that? That would be extremely useful if you can think of things. Because often we didn't know what to do in this hour. So if you can help us by telling us, before this, before you teach this, teach us that. That would be extremely useful. Okay, let's go over the syllabus.
I have some delay. We didn't learn we learned about eigenvectors, but not about left and right, and for me this was confusing at the beginning.
Yeah, we did study it in the focus hour, but after you introduced it in class, then we were...
What about the linear algebra course that is given?
But you just learned eigenvectors, not like left and right.
You did not learn in the linear algebra course. You don't learn left and right eigenvectors.
No.
Can you?
I think they only get to eigenvectors primarily in the end.
Yeah.
Well, what else is there in the linear algebra?
You know what's the matrix?
What's it called? Row echelon?
Row echelon.
Row echelon form. Right?
No, but they do.
I've talked to them about it, but it doesn't have any changes.
Okay, so this is something we need to write down and discuss with the teachers next year. More things? Okay, let's talk about the thing that we did and the thing that we did not do. So we spoke about the rate model of neural networks, dynamics, working memory, feature selectivity, associative memory, rate cards and asymmetric networks, irregular spiking. We did not talk about decision making and perceptual rivalry, which is perhaps somewhat sad, but we spent more time this year on balanced networks, and we spent more time on associative memory. So no time was left for other topics, but perhaps it's not such a bad thing, because there are so many things on the list that we also did not do. So the fact that we did not do these two, this is not a reason to be sad. Because the main goal of this, for me, the main goal was to teach you methods in the way of thinking, which is applicable also to not only to computational neuroscience, but also to computational cognitive sciences. This would be nonlinear dynamics, stochastic processes. They appear also there. So whether or not one specific topic or another is taught, it's not that critical. I would like to spend the last 10 minutes to discuss several thoughts about modeling in neuroscience, more generally. So what's the use of mathematical models in neuroscience? And people do neuroscience without mathematical modeling and have been doing it for I don't know, half a century at least. And people have been doing electrophysiology, measurements. So what is it that we gain from these mathematical models? And I think that there are some things that we gain, and at different levels. So one thing that we gain is it provides us with a sophisticated way of analyzing data. Sounds like something that is not a lot, but for example, when we talked about balanced networks and we talked about computing the CV as a measure of irregularity of the neurons, I don't think that people would have used this measure if not for thinking mathematically about the problem. So by introducing models, we also introduce more sophisticated ways of looking at our experimental data. So that's just another measure. Rather than looking at, I don't know, the average, we also look at other things. So this is one thing that we gain from modeling. Another thing that we gain from modeling is that intuition takes us some way, but I don't think that we can understand things like an action potential, talking about the single cell level, or the irregular firing in cortical networks and the relation to the distribution of firing rates without a mathematical model. That's my view. I mean, this is here. You have to use a mathematical model. A third place where we use modeling in neuroscience, and this you do more next year, is we often use functional models. And this is a completely different approach. So when we talk about dynamical models, in some sense, explaining the cortex or explaining an action potential is not fundamentally different from understanding the spatiotemporal structure of radiation coming from the sun. This is a dynamical system. It has particular properties, and we're trying to understand these properties. So this is part of, let's call it normal science, or maybe science. When we talk about functional models, this is something completely different. Here what we do is we think about the brain as a machine that computes things. So this is where neuroscience is special and different from physics. So we think that the brain is computing, and often we think about what's the optimal way of solving a task, what's the optimal way of performing a computation, and then we compare these optimality considerations, we compare them to what we think that the brain actually does, or we go to our friends in the department of computer science and we tell them, look, we have this problem. No, I don't know, we want to recognize faces, what should we do? And they tell us do this and that. And then we ask them, okay, what would be a signature of this algorithm? And then they tell us, no, we expect this and that, and then we go and look for it in the brain. Yes? So it's related to biology in the sense that it assumes that we have a function.
So you just
We have a goal in life.
but all the ingredients can be completely different.
What do you mean by ingredients?
I don't know. At the end, you get a certain function, but maybe you can get it with different components.
What's
But-
the difference between this and computer science?
First, we assume that there is a function. Then we try and figure out how this function can be achieved. And then we look for signature in the brain that will relate the dynamics of the brain to this computation. This is what we do. This is what you would do. You spend most of the time in the next semester. And there's I think, a fundamental difference between these two approaches. This, in some sense, is much more interesting to us as neuroscientists, because there is a reason why we study the brain. We don't study the brain because of its irregular spiking. We study the brain because it's interesting. It's, we are the brain or something. Because we think that the brain does interesting things. So this is what's special about the brain. So this is why this is so interesting. Of course, in this course we did this. I even have colors. Okay, so we did this. I don't know about the sophisticated data analysis, but we did mostly this. And this is, in some sense, the more neuroscience special thing, or the biology special thing. You also do it in, I don't know, you study your favorite muscle, and then you say, okay, the goal of the muscle is to, when contracting, to exert force, and this is why it's built this way and not another. So this would be an example of a functional approach. The advantage of this approach over this approach is that this is real. The brain is a dynamical system. There is input and there is output. So this is science. Okay? This is more complicated. There are assumptions here. There are assumptions about evolution. We study the eyes, and we say, okay, we evolved to find prey and escape predators, and this is why we need to make these computations. We need to be sensitive to motion, and indeed we see that the eye is sensitive to motion, and so on and so forth. So here there are many assumptions, and eventually this will break down, because this is reality. This could be a very good approximation, but this is reality. We have to bear this in mind. Now this is the interesting part, in some sense, at least interesting to me, but this is reality. And this underlies this. So when you think about modeling in neuroscience, I think it's worthwhile thinking about the difference between this and that. Any questions? Any remarks? Yes?
Is there any example that you tried to study a function using these function models and then you found it in the brain?
Well, of course. That's what much of neuroscience is done today. So single cells, action potentials, is no longer, let's call it, people don't no longer find it too interesting. The reason is that we pretty much understand how action potentials are generated and the role of photonic channels. So if you have a particular problem, then you may want to study this particular. But in general, at least we have a feeling, I don't know if it's true, that we understand this. This is more challenging. Now, this is what, if you go to cosine, mostly in those talks about dynamics, you'll hear talks by, Itamar presented in cosine a year and a half ago, I think, talking about his model. So these are the kind of things that people now are interested when people talk about dynamics. This was not true 15 years ago. 15 years ago, you'd see many papers like this. Now, what we often do today in computational neuroscience, you have your favorite computation. I don't know, Tell me what you are interested in. Something. Love. Okay, love. Okay, so what you do an experiment about, you have two conditions. You do humans. You present the person that you love against the person that you don't love, and you try to identify, using fMRI, a brain region which is more selective to someone that you love than someone that you don't love, and you'll call it, this is a love region. And if it would be even better if you love sushi. So you'll have sushi versus pizza. This would be the contrast. You see, orbitofrontal cortex, and then you see, your spouse versus your enemy, and then you find a localization of activity in this region, and you say this is a love region. And the idea is that love is important because we value things, and eventually, this will derive from this. So there's a lot of neuroscience on function. That's a lot. I think most of computational neuroscience today, this is what people do. Because this is, in some sense, this is what's special about the brain. The fact that we do computations.
How can you infer from your model to neurology? So what can you say? Okay, there's a model and it does this function, and we know that the brain does this function, but what do you learn about the mechanism underlying this phenomena in the brain?
So for example, one example would be inference. You have information coming from two sources. How do you combine two noisy sources of information? There's an optimal way of doing it. There's an optimal way of calculating it. And you look for, first you look in behavior whether this is indeed how humans or animals do it. And then you'll try to find, you have an algorithm how to combine these two pieces of information, and you look for a brain region that is selective for different parts of this calculation. That would be one example. A second would be to ask a question. Let's assume that the brain represents stimuli in a particular way. What would be the best way of extracting the information from this noisy representation in the brain? And now you can compare the actual behavior of humans to the predicted by optimality considerations. This is kind of thing that people would do. Yes?
You draw a line here between the dynamical and the functional model.
Yes.
And how much is the area around this line, like the combination between the two, studied, or can it be studied, or is it too far away now?
No, of course it can be studied. For example, people can ask a question. We spoke about the Hopfield model for associative memory, and you can ask the question, is it possible to store memories in balanced networks? What would be the constraint? What do you need to assume to get multiple fixed points in balanced networks? That would be one example. If you talk about the periphery or the retina, then people do a lot. So you can say, for vision, it's useful to find edges. Edges are in order to identify edges, you need a high-pass filter. How do you construct a high-pass filter? You go to the retina and you look for interneurons that do some kind of lateral inhibition that results in dynamics, which is high-pass filter. So this would be an example of combining the two approaches. Now, don't get me wrong. I'm not saying that this is not a valid approach. I'm saying that we should be more cautious here than we should be here, because this is treating the brain like a dynamical system. This we know that the brain is a dynamical system. This is introducing additional assumptions, which can be. It's kind of stupid to study the retina without knowing that it's useful for vision, right? So this would be an example of introducing functional considerations when studying biological tissue. And again, most of what we did here was to talk about dynamics, although we discussed how computations like feature selectivity, working memory, associative memory can emerge from the dynamics. Questions, comments? I have to teach in 10 minutes, so I have to run. So thank you all, and see you in the exam.